Welcome to this Antibody 101 video series. Our goal is to highlight the high-level concepts for antibodies in particular and proteins in general. My name is Kathy. I have a PhD in synthetic biology from Stanford. I trained as a postdoc in computational protein engineering at the Institute of Protein Design, and I've worked as a scientist for many years in big biopharma. In this video, we'll cover Antibody 101 Part 2, which is how antibodies can kill cancer. So this image is an SEM, or scanning electron microscopy image, of a cancer cell shown in green on the right here, and a natural killer cell shown in orange in the left here. So I'm showing you this diagram just to show what these cells actually look like under a microscope. Now, if we look at the diagram version of this figure, we have the cancer cell in green on the bottom, and this natural killer cell in orange at the top. And you can see that in between them is an antibody. And this is one of the ways that an antibody therapeutic can work. So if you'll recall from last time, the antibody is like a lobster with two lobster claws. And these claws essentially can attach to some bit of the cancer cell that is sticking out. And that part we call the antigen. Now, the antibody body, or the lobster body, as you can think of it, has another function. And one of its functions is to be able to attach to the surface of, say, a natural killer cell. And the result of that is it brings the natural killer cell into very close proximity with the cancer cell. Now, why do you want this? Well, the result is that the natural killer cell will actually kill your cancer. So this is one of the many ways that an antibody therapeutic can work to defeat cancer. So let's zoom in a little bit. One particular cancer antigen, aka a bit of protein that sticks out on the outside of a cancer cell, is HER2. So some of you might recognize this name from breast cancer, where it's kind of a famous target. Now, HER2 is a protein, and it has three different parts. One part of it sticks outside the cell. This is called the extracellular component. There's also another part of it that goes through the membrane. And if you'll recall from your biology classes, the cell membrane is what defines the inside versus the outside of your cell. And then the third part of this protein is intracellular, or it is inside the cell. And this is a part that, in a regular circumstance will signal to your cell to do whatever the natural function is. Now, for the purposes of antibody therapeutics, we only really care about the extracellular portion. So this part of HER2, this protein that sticks out on the outside of your cancer. In this diagram, you can show that it's broken up into 4 domains, 1, 2, 3, and 4. And there are marketed drugs that are antibodies that actually can bind and recognize different domains on this HER2 antigen. So pertuzumab is shown here, and trastuzumab is shown here. As a side note, most antibody therapeutics have many names. For example, pertuzumab is marketed under Perjeta, and trastuzumab is marketed under Herceptin. You'll actually see different names for different countries, for example, or for different indications. So it can be a little bit confusing. So there are multiple names for the same exact drug. Okay, let's look at this in a little bit more detail. On the left, again, is the diagram we saw from last time. Gray is HER2, the protein antigen that we want to target, and trastuzumab is the antibody in yellow, and pertuzumab is the antibody in this red-pink color. So if we look at the actual structure of the protein in this surface representation, we'll see that this is kind of what HER2 looks like in gray, and pertuzumab looks like this pink and red blob here, and trastuzumab looks like this orange and yellow blob down here. And you can see that they do actually bind HER2 in very, very different places. So if we zoom in for even more detail in this cartoon representation, you'll see a little bit more of what exactly makes up HER2. We've got some sheets here and some helices here. And you'll also see the details of the sheets of both pertuzumab and trastuzumab. And just to give you guys a sense of the sizes of these things, HER2, just the extracellular domain, is about 630 amino acids. Remember that a protein is made out of a chain of amino acids, and in this case, there are 630 of them stacked together. And then these Fabs also have a size. So a Fab refers to the part of the antibody that is essentially the crab hands of the antibody lobster. So a Fab is approximately 430 amino acids. Okay, and just to zoom out a little bit, again, what we saw in the last picture is just the crab arm of the antibody. And now we can see that the full antibody takes up this sort of shape here. And this gives you a better picture of what the full HER2 plus an antibody might look like. Okay, just as we did last time, let's zoom in on the very tips of those lobster claws. So instead of the entire claw, we're actually looking at just the very, very pincer tips of it. And if you'll recall from last time, these are called the VH and the VL domains for variable heavy and variable light. So what we've done here is superimpose the structure of both pertuzumab and trastuzumab together, and that is what is shown above. And we can quantitatively measure how well they overlap with something called a TM score. So a TM score of 1, you can think of as 100% identically, perfectly aligned structurally, and a TM score of 0 means there's absolutely no alignment between the two. At about TM score of 0.5, that's typically considered the same fold, so the two proteins are, roughly the same shape. And usually at TM score of 0.8 or better, that's considered very, very close. So in this case, the point is, even though these bind to completely different places, structurally they are incredibly similar. And this is very common for antibodies. The other sort of alignment or comparison we can do, aside from structure comparison, is sequence comparison. And I'm showing the exact sequence alignments of the VH on the bottom and the VL at the very bottom. And so if you look at the sequence identity numbers between these two sequences, the VH is 75% identical and the VL is 89% identical. Again, the point here is to illustrate that for an antibody, the sequence IDs can be extremely, extremely high. So very, very similar, but functionally they're very different. As we saw on the previous slide, they bind to completely different places on HER2. Okay, so in conclusion, just like these screwdriver heads can only be a tiny, tiny bit different, but functionally they matter a lot. As with antibodies, even if the sequence or the structure are very, very similar, their functions can actually be very different. With that, thank you for learning with us today. Biology breaks every rule that it makes, so remember that the ideas presented here are one way, but not the only way that things work. Please like and comment, and see you in the next video.